Next speaker is Koen Dechering. He's from TropIQ, the Netherlands, and a spin-off company of the Radboud University. And he will tell us about scalable assays.
Thank you, and thanks to the organizers for inviting me. It's my pleasure to talk to you about our efforts in implementing and validating a screening cascade for prioritization of transmission blocking antimalarials. And we are particularly interested in predicting infectivity of gametocytes. And gametocytes are a bit like the iris setter depicted on the cartoon here. It's quite difficult to tell in what particular mood they are. So whether they are happy or depressed. It's even very difficult to tell whether they are dead or alive, let alone can we predict they're infectious to a mosquito. So we're interested in identification of markers that predict infectivity and to look at the quantitative relationship between the expression of that marker and infectiousness of a gametocyte. So what does it mean if you inhibit the expression of a luciferase reporter gene by 50%, or what does it mean if you inhibit the number of X-flagellation events by 50%? Does that mean that you have 50% less transmission, or is that correlation more complex? So we looked at two markers in gametocytes. One is PLDH, just explained to you by Pietro, and work from Donatella Taramelli's lab has shown that gametocytes express very high levels of PLDH. So we looked at PLDH in an assay format that we slightly modified from the colorimetric assay that has been described before. So we use a fluorescent substrate which gives us better sensitivity in the assay. We also look at the transition from gametocytes to gametes by looking at PFS25, which has been introduced by Michael Dels. We use a different technology though, so we use two antibodies that recognize PFS25 using non-overlapping epitopes, and both antibodies are coupled to alpha beads. And if you excite one bead with a particular light of a particular wavelength, it produces an oxygen signet that then excites the other bead. And that only works if they are in close proximity. And the beauty of that assay is that it works in solution, so there's no washing step. You just throw in the beads and you can measure the PFS25 level. So both assays run in 384 well plates, and they're quite robust as we see Z prime values that are above 0.7. So we used these assays to look at the MMV validation set, a set of 48 compounds, mostly marketed antimalarials, a few experimental compounds. And here you can see a percentage inhibition when compounds were tested at a concentration of 5 micromolar. And on the x-axis is the PLDH inhibitory activity, and on the y-axis the inhibition of the capacity to form a female gamete. And they correlate quite well, and you should bear in mind that PLDH looks at the whole population of gametocytes, whereas PFS25 only looks at the females. Yet it appears that the PFS25 assay captures everything that we see with PLDH, and even more, as there's a couple of compounds that do show activity against the gametogenesis capacity, whereas we do not see them in the PLDH assay. That these assays correlate quite well is also shown here, where we compare IC50 values, and when we plot them, you can see that the IC50 that we get on PLDH versus the activity that we get on gametogenesis capacity, they line up pretty well. So a reduction in PLDH seems to go hand in hand with a reduction in female gamete formation. We use the female gametogenesis assay also to look at the kinetics of compound effects, and we tested compounds by incubating gametocytes for either 72, 24 or 1 hour prior to activation of gametogenesis by addition of centuronic acid, and then we measure PFS25 16 hours later. And it appears there's 3 types of responses. An instantaneous response where the expression of PFS25 is almost instantaneously prevented. An intermediate response where the steepest part of the curve is in the first 24 hours. And a slow response where the steepest part is in the area of the curve between 24 and 72 hours. And this seems to be a class effect, as the immediate acting compounds are 2 translation inhibitors. And the intermediate effect is mainly dominated by endoperoxides, whereas the slow acting compounds are mainly 4 aminoquinolines. So we were then interested in how these PLDH activities and PFS25 effects translate in transmission effects. And so this is the standard membrane feeding assay, and analysis is based on the dissection of midguts and microscopy, which you might appreciate is very labor intensive. So we've been looking for ways to improve throughput in this assay, and for that we developed a reporter parasite that gives a luminescent signal in the mosquito midgut. And the approach we took is very similar to an approach that is presented by Will Stone on a poster that's on display today. But Will looked at a reporter parasite developed by Stephen Kappe, which uses a reporter driven by EF1 alpha promoter, whereas we generated a novel reporter strain that expresses GFP luciferase from a HSP70 promoter. And the selection cassette that was used was flanked by a flirt size, so we could transfect the parasites with flip recombinase and excise the selection cassette. And the end result is a reporter parasite that's free of all selection markers, so it's fully sensitive to all drugs, including antifolates. So we use this reporter strain that expresses the reporter kind of throughout the life cycle to look at intensity of infection in mosquitoes and compare luminescent signals to microscopy-based readouts. Both assays are destructive, so we can't do that in the same mosquito, but we analyze mosquitoes from the same cage with microscopy or luminescence assays. And we analyze over 170 cages of mosquitoes. And here you can see the correlation in oocyst intensity as assessed by microscopy versus the luminescence signal that we get. And here's the correlation for prevalence, or the number of infected mosquitoes. So mosquitoes harboring at least 1 oocyst. And you can see that there's a good correlation between the two. And we can pick up a single oocyst by looking at luminescence signals. The assay has reasonable throughput, so we can put mosquitoes in microtiter plates. So we put them in 96 well plates and add ceramic beads, then shake them in a bead beater, add the luciferase reagent, and analyze 96 mosquitoes on a plate. So we use this luminescent SMFA then to interrogate this validation set. We pre-incubated gametocytes 24 hours with compounds prior to feeding to mosquitoes, and then looked at luminescence intensity 8 days post-feeding, and we analyzed 24 mosquitoes per sample. And you can see that we identified a considerable number of compounds that blocked oocyst development in the midgut. So now when we compare this data, so 24 exposure of gametocytes to the data that we generated in the PFS25 gametogenesis assay, you can see that the SMFA, which is here on the y-axis, is much more sensitive than this gametogenesis assay. So apparently gametocytes lose infectivity before they lose the ability to express PFS25 on a gamete. That seems to be a bit of a delayed response on the gamete, because when we compare the SMFA data to the 72-hour incubation in the gametogenesis assay, you can see that some of the compounds shift to the right, so they better reflect what we see in the SMFA. So part of the discrepancies that we see, so compounds that are not seen in the gametogenesis assay but that do inhibit oocyst formation, might be explained by sporontocidal activity of compounds. So when we do the SMFA, we do it in what we call the indirect mode. So we pre-expose gametocytes to a compound, and then the compound remains present in the blood meal, so it's there in the mosquito midgut. So we tested compounds that were active in this particular mode of the assay also in the direct SMFA, where we add the compound at the moment of blood feeding, and that would reveal any activity that is downstream of the gametocyte. And here you see luciferase intensities normalized to the controls, and the open bars are the activities in the direct mode and the solid bars in the indirect mode. And you can see that many compounds are not active in this direct mode, but there's 3 classes of compounds that are acting in the direct mode, so that might have sporontocidal activity. And these are the 2 amino alcohols, the antifolates that we tested, and the single BC1 inhibitor etovercona that was tested. So we can explain the higher sensitivity in the SMFA for transmission blocking compounds partially by the fact that some of these compounds are sporontocidal and not gametocytocidal. But still there's a couple of compounds that are not picked up by the gametogenesis assay, and also not shown on this slide, but they are also not seen in the PLDH assay. So we validated a number of these compounds in more extensive dose-response assays, in membrane feeding assays. And the results are shown here, again comparing microscopy to luciferase measurements. So luciferase is in red and microscopy is in blue. And you can see that the data align very well. So indeed, luminescence reflects what we see in microscopy. And we could also confirm that these compounds are transmission blocking. So etovercona and pyrimethamine that are sporontocidal, but also 2 compounds, lumefantrine and mefloquine, that are not active in the gametogenesis or PLDH assay, are transmission blocking when we test them in the SMFA. So, in summary, when we take the SMFA data as the ground truth and compare the data from the other assays to this set of data, we conclude that PLDH and PFS25 assays have no false positives. So everything that lowers PLDH or PFS25 expression will pop up in the SMFA and show transmission blocking activity. However, there's a pretty high false negative rate. So these 2 gametocyte markers that we looked at miss some of the compounds. And in this analysis, we excluded the sporontocidal compounds. So there are compounds acting against gametocytes that we miss in those 2 gametocyte assays. And I think that indicates that the set of functions that describes gametocyte infectivity is larger than PFS25 and PLDH activity alone. So where do we go from here? We are currently testing a larger set of compounds that have been identified in gametocyte assays by different groups all over the world. And we test them in the SMFA, in the luminescent SMFA, in dose response to provide a reference data set to this community. But we are also aiming at increasing throughput in the SMFA further, as it seems to be more sensitive than these 2 gametocyte assays. So we are trying to develop an SMFA with even increased throughput, so we can put it up front in a screening cascade and really use it as a primary selection assay. And for that, we are developing technologies that would allow us to feed mosquitoes on 96 well plates, where every well would have a different compound. Well, the challenge then, of course, is how do you track which particular mosquito has fed on what particular well. And to that end, we put a barcode in every well, and that barcode is a endosymbiontic bacterium that colonizes the mosquito midgut, and we can transform the bacterium with a plasmid harboring a unique DNA sequence. And the idea then is that we screen for mosquitoes that have no luminescent signal, so they have been feeding on a compound that is transmission blocking. We can then rescue the bacteria from the mosquito midgut and analyze the DNA barcode sequence content that we get from that particular pool of mosquitoes. That is all pretty much work in progress. There were a couple of challenges, but we have established proof of concept, and this is showing an experimental setup. So we get efficient feeding on 96 well plays. We get coverage of approximately 5 mosquitoes per well, and we can retrieve barcode sequences from fed mosquitoes. And we are now in the process of scaling up this technology and increasing the set of barcodes so we can start interrogating a larger set of compounds. With that, I would like to end my talk and acknowledge the wonderful team at TropIQ, our sponsors, and also our collaborators in the Gametocide consortium that we are part of. And happy to take any questions.
It's hard work in Nijmegen, as you can see. It's very dangerous and bloody. Any questions? Yes. What? Yeah. Shout or go to the microphone, please.
Yeah, that's a good question. So the question is, what happens if a mosquito feeds on the multiple wells? So the observation is, and so we put a camera in a mosquito cage to film feeding behavior. And so the particular laboratory colony of mosquitoes that we used, so once they hit the blood, they tend to fill up and not probe on different wells. So around 10% of the mosquitoes does show cross-feeding behavior. But since we're aiming at a sampling of every well with multiple mosquitoes, it will be very unlikely that all mosquitoes will have the same cross-feeding behavior. So there will be low contamination of cross-feeding.
Yes.
I was just also curious about the timing of this, because if you have a gametocyte that's living for, 10 days and then 4 or 6 days afterwards, yet the drug has to act only on the stage 5s. You've got a whole lot of gametocytes that, unless you keep your drug pressure on, are going to, as soon as that, the drug declines, then the 2s that were there, the 3s that were there and weren't affected, are going to now be infectious. So you're going to have a very long course of treatment. True? So how do you see this kind of fitting in with your assay and making these decisions?
Yes, so most of the compounds that are chazontacidal also affect the early stages of gametocide. So when the drug is there, they probably get rid of all the developing gametocides. But there might indeed be a window where you have more advanced gametocides progressing
10 days
into stage 5.
is a long time.
Yeah, I think it's a fair point, yes.
Yeah. So, Koen, with maybe a little bit provocative, but with all these false negatives and the previous presentations where we trying to identify, obviously, compounds or leads, how do you see the pipeline?
Yeah, so people that are in high throughput screening, they know that if you use a different technology, you get a different answer, right? So if you have a target that you're really interested in, you would use all the technologies available to increase chemical diversity in the collection of hits that you get.
So that, yeah.
But I guess at some point you might saturate. And I think we need to be looking at the collected set of assays that are available and generate this SMFA reference data set and see where the gaps are and how much coverage we get. Yeah.
So there's also some discrepancy about what Michael said when the sensitivity of males is, there are particular compounds there. Well, one of your statements was that you actually pick them up and that for using a female assay you don't lose anything, apparently. So they are not male specific, or how do you see those compounds?
Yeah, that is puzzling. So we're using a different technology, so it might relate to the different technologies that are used, or it might be in it might be in the timing that so that females have a bit of a delayed response And Michael, remind me, how long do you incubate?
It's only 24 hours.
Yeah, so maybe females have a bit of a delayed response, and you don't see those compounds, and we do see them when we incubate for 72 hours.
I think, like Koen says, I mean, anything inhibits anything if you give it high enough concentration and enough time to act. And so I think in our assay we've got a shorter exposure period, and males are more sensitive than females, so that doesn't mean that it's not active at females if you give it a high enough concentration or leave it longer. And some of the tests we've done, if we incubate for 48 hours, some of the compounds which do look male specific at 24 start to come up as active as females as well. So I think it's an exposure thing.
Exposure thing. Okay. Thank you very much.